Good morning, good afternoon, and good evening. I'm going to be talking about visible spectroscopy. My student number is 2020327. An introduction to ultraviolet spectroscopy. The absorption of ultraviolet and visible radiation is primarily known to be electron excitation in atoms and molecules through a lower to higher energy levels. These energy levels, if higher, the more wavelength is being absorbed. The electromagnetic spectrum. Again, I can only apologize for my poor drawing, but I will be talking about this further in the next slide. The electromagnetic radiation. It can be defined as a number of electrical field radiation oscillating in 1 second. To measure it's in hertz, 1 hertz per cycle per second. The wavelength, this is the distance between two parts that are nearest of the wave, which is the same in the same place. Visible spectroscopy. Seeing color is a sense of which happens when light enters the eyes and it inhibits the retina. This process inhibits a process called photochemical, which is carried through the nerve cells in the retina, which aid in transporting these messages to the brain and create our reality of color. To break this down in very simple terms, cones, they give out color, of which are red, blue and green. Rods, gray and black, are given. These colors that we are made, that we see are made up of these colors. Visible spectroscopy. White light composition. Sunlight is a white light with a wavelength range of 380 to 750. White light is proven to be composition of a range of colors, so it's a light of different energies and therefore wavelengths. The absorption/reflection properties of these materials in an object determine the color of object when light falls on it as detected by the eye. Complete reflection of a material appears white. Constant fraction of light across the spectrum absorbed by a material appears grey. Complete absorption of a material appears black. And these are the images of the lab, and my understanding was that there was two parts. One showing a bit of solution into the machine, and another to do with food dyes and colouring. So from the lab results from these both experiments, they were nil as I was not present for these both experiments, and those are my reference. Thank you.
Very good.
Round of applause, man. A round of applause.